So I am Tom. I'm a postdoc here at MIT, and today I'm going to be talking to you about scaling AI-enabled protein engineering. And for this, I think it's important to focus on the context I've been working in, and particularly the application spaces. So if you join me here in the middle of this design, build, test, learn cycle, I've been working lots on neglected tropical disease, and particularly developing recombinant snake antivenom, and also in bioindustry and sustainability, so discovering binding proteins that would modulate the activity of industrially or agriculturally relevant enzymes. Now, these seem like two fairly disparate fields, but I do believe they are united by a commonality, which is that lots and lots of our extremely powerful modern biotechnological tools could be applied to solve a vast range of problems in these spaces, but we are limited by the costs associated with implementing those tools. So my thesis is essentially that by reducing costs, we can increase the number of people that are working on relevant problems, and thus increase the number of solutions that are found. To reduce costs, I believe we can kind of combine lots of advances we've had in these three themes, and particularly relevant to today is in the design space for using phenotype-enriched library design. And this can either be kind of taking notice of nature's ability to design things, so looking at metagenomic databases, using AI-guided directed evolution, or one of my favorite things now is just entirely de novo protein generation. And by combining these things, I think it becomes possible to say that we can achieve things like this, where we can get 1 to 10 binding proteins against a specified epitope on a target protein with in vitro validation and in silico structural prediction for less than $100. And hopefully you agree with me that that is a very impactful price point if we want to actually go out and develop solutions in some of these spaces. So to talk you through why I believe that this is a reasonable statement to make, we can start with our work in the design space, which was through a collaboration with the Baker Lab using RF diffusion to de novo design mini binders against snake three-finger neurotoxins. With this, we achieved a 2% hit rate with picomolar to nanomolar affinities, and most importantly, achieved functional in vivo neutralization of these toxins. And this was massively aided by the ability to pre-specify the epitope on the target that we wanted to bind to, so we could block the known toxin receptor interaction in vivo. If you want to learn more about that, you can follow this QR code in the bottom left to the preprint. So after that work, what quickly became abundantly clear to me was that design was no longer a particularly rate-limiting step in our ability to implement solutions, and instead that fell more on our ability to build designs and actually test them in the lab, and this is something touched upon already, the ability to generate data or experimentally validate our in silico predictions. So for this, just as an example, I've shown here how much it costs to actually assemble different numbers of 400 base pair mini binder genes depending on the vendor, and this is going from ordering the DNA to having an entirely sequence-validated clone. This, I think, is best showcased with this example of scaling it up to test, for instance, 3,000 designs. If you order this as e-blocks from IDT, it costs over $80,000. From Twist as multiplex gene fragments, it's about $25,000. But you can use alternative sources of DNA, so for instance, oligopools, where you can get the cost down to about $3,000 to $4,000 for this, or about $1 per design. Now, to actually achieve this, you need to assemble the oligopools into full-length genes yourself, which I've built a pipeline to do in the lab, and this I can chat about more at my poster if you want to come and talk about it. But just to briefly show that we're already, just on our first pass of this, achieving 76% of assemblies being entirely error-free without any required error correction processes. Obviously, all of our building means nothing if we can't also test. So for this, we've been establishing the use of split luciferase, and particularly in a kind of automated setup, where it costs about 6 cents per assay to test each design. And with this, we can get the expression level of a protein of interest, and also the binding affinity and specificity. And what's very nice about this is that it's an entirely no-wash homogeneous assay. It works in cell lysate, so it's very simple to implement. We're now using that to test more designs against snake three-finger neurotoxins, and in particularly using the scalability of this pipeline to look for cross-reactivity, which we believe is going to be a fundamental phenotype for achieving an actually cheap and effective snake antivenom product. So hopefully today I've interested you in maybe some new application spaces of lots of the tools that we're developing, and also conveyed to you the importance of building more efficient build and test cycles. Thank you.